Okay. As at yesterday, South Africa has recorded more than 2.8 million infections and more than 85,468 deaths. And globally, more than 290 million cases of infections have been recorded and over 4.5 million deaths already. This is getting serious. Good morning, everyone. Today I will coordinate the program. I will coordinate the program. And just before I start introducing everyone, just some house rules. Please, for those on Zoom, kindly turn off your camera and mute your microphone unless you are participating in the question and answer section. Please type your question in the chat box during the course of the program. Then I would like to also acknowledge the presence of the following people. Number one, Professor Medupe, our Deputy Dean for Community Engagement and Impact. Professor Carlos Benzidelt, our Unit Director for Environmental Sciences and Management. The subject group leaders that are here today, Dr. Lexeko Mulalitom, Dr. Ezel Mufandu as well. And the members of the organizing committee, I don't want to take over the job that Prof. Carlos will eventually do today, so you'll mention your names later. And also acknowledge the presence of all other colleagues that are here today, as well as all the students that are in attendance. Let's assume for a minute that this COVID-19 that we are talking about is a social media influenza. Definitely, it's obvious that it will have most followers by now. So it is only reasonable and logical for us as a department to focus on this topic today, so as to celebrate the World Microorganisms Day. So of course, there could not be any better discussion than COVID-19. It has to be COVID-19 today. So you are all welcome once again. So we will start by inviting Professor Rodney Medupe to give a welcome address. So over to you, Prof. Medupe. Thank you.
Thank you, Program Director, Professor Adeleke. Distinguished guests, fellow academics, students, ladies and gentlemen, good morning to all of you. On behalf of the Faculty of Natural and Agricultural Sciences, we welcome you all to today's event. A very special warm welcome goes to the main speaker of today, Dr. Jinal Beeman. We are looking forward to learning a great deal from you today. Let me also acknowledge the presence of Professor Carlos Bezaidin-Hout, who is the Director of UESM, Professor Sarina Claessens, who is the Director of the School of Biological Sciences, and Professor Olubukula Babalola, who is the Director of Food Security and Safety. Also, welcome to other colleagues who are present today, especially to Professor Adeleke, Dr. Lexeko Mulalitom, and all other colleagues that I have not mentioned. International Microorganism Day is a day in which we celebrate Anthony van Leeuwenhoek, the first person to report and describe a single-celled organism 338 years ago. His discovery formed the basis of modern microbiology, which is why he is known as the father of microbiology. Now, out of sight is a common phrase that is used in everyday life. However, most people aren't aware of the complex world of microorganisms or the impact that these organisms have on our lives. Thus, International Microorganism Day, IMD, shines a light on these unseen organisms and brings light to the impact that these organisms have on our daily lives. International Microorganism Day allows researchers to share information and acquired knowledge of microbes present in our daily lives. Yearly, the Northwest University Microbiology Department commemorates International Microorganism Day by sharing with the public at large the research conducted within the subject group. The research conducted within microbiology is diverse and multifaceted. The Potchefstroom campus researchers have ventured into the field of microplastics and are conducting research on how plastic particles are vectors of antimicrobial resistance in the environment. Research is also being conducted on escape pathogens, which are rapidly growing multi-drug resistant organisms prevalent in hospital settings and recently showing up in environmental water systems. Another aspect of the collaborative research focuses on the isolation of endophytic microbes, which have the potential to improve crop yield. Research is also underway to investigate the use of microorganisms as biocontrol agents in agriculture. At the Mahikeng campus, research is being explored on the usage of bacteriophages in the fight against antimicrobial resistance. The healing properties of medicinal plants and compounds produced by the associated microbes are also being studied. While food safety and security are also being explored. These are some of the contributions made by the Northwest University to better understand the way microorganisms function and their role in human existence. In the past, not a lot of time was spent by the public reading up on microorganisms and the impact that they may have on public health until last year when all our lives were changed by COVID-19. This single-stranded RNA virus is encapsulated in a studded capsule of glycoproteins. This virus is highly adaptable, and the repercussions of the rate at which it is spread are felt right across the globe. In recent times, the focus point globally was for the development of a vaccine to save lives. There are various vaccines available, and the two main vaccines available in South Africa include Pfizer and Johnson & Johnson vaccines. Once again, the Faculty of Natural and Agricultural Sciences, together with the Department of Microbiology, welcomes you to today's events and wish you to enjoy and learn from the main lecture or webinar, as well as the discussions that will follow. I thank you all, and in particular the organizers as well. Thank you very much.
Thank you very much, Prof. Rodney. Very insightful statements there. I'm sure you've read her CV. She's a researcher, but as we know, she's a good researcher anyway. And we know that a good researcher must be hardworking, diligent, focused, and devoted. These are all reflected in the CV of our speaker today. She has published widely. This year alone, she has published more than 10 articles, 4 of which are in Nature journals, 2 in New England Journal of Medicine. You know that these are top journals. These are not just publications. She has contributed a lot to the research fields of HIV and the COVID-19 pandemic issues. So, but one thing that is clear with our speaker today is she's somebody that is a go-getter. She has worked extensively on HIV vaccine, but recently her devotion in the last 12 months has been on the COVID-19 research. I am sure you've gone through so many of the other achievements that she has in her CV. So ladies and gentlemen, it is my pleasure to introduce one of the greatest, one of the most vibrant researchers that we have in South Africa today, the director of the National Influenza Center and RSV Reference Laboratory, Dr. Gina Beeman. Over to you, Doctor.
Thank you, Prof. Rashid, for that very flattering introduction. Let me just start to share my screen with you all. Okay, so thank you to all the distinguished guests. Thank you to the organizers of this wonderful program today for inviting me to give this presentation. And thank you to all the attendees, to all the students. I really hope that this is going to be beneficial to you and that you'll learn something, and that the Q&A session afterwards will answer some of your questions. So the topic that was given to me was coronavirus and vaccine perceptions. And I just want to put a disclaimer out there right now. I'm more of a virologist, and especially with respect to SARS-CoV-2, I've really been focused more on looking at the evolution of this virus and looking really at how those mutations that have arisen are impacting its function, impacting its transmissibility and escape from immune responses. But of course, you cannot be in this field without paying attention to vaccines. So I'm hoping my knowledge of the coronavirus vaccines will be good enough and will really answer your questions. So just a very basic background, which I'm sure most of the students here already know. SARS-CoV-2 is the virus which causes COVID-19. And I've got here a little schematic which shows, actually maybe I should use the laser pointer. Yeah, so I've got a little schematic over here which shows how the virus kind of looks. It's got a lipid membrane, and embedded within those that lipid membrane are these little spike protein, and then within the virion is the nucleic acid, which is RNA. It's got a relatively large viral genome of 30,000 RNA letters, or 30,000 bases as we know it. And this is now a schematic of those 30,000 bases, which encode for quite a number of genes, but only 4 structural genes, which I already mentioned. The spike protein is coded by the S gene. We've got an envelope matrix and nucleocapsid gene as well. And then within these 2 large polyproteins, there are many little accessory genes, and also the enzymes that are required for replication and packaging of the virus. So as I mentioned, this is now electron microscope image of the virus. So you can see very nicely these little spikes that are sticking around outside the virus. And the spike protein is really what is the major target for vaccines currently. And I think that kind of is intuitive and it makes sense. It's found on the outside of the virus. And so really when this virus infects you, that's the first thing that your body will see, is what is on the outside. And that's why this protein has been selected for as a vaccine immunogen. So just diving into a little bit of the molecular structure of this spike protein. So it is a trimer. It's made of 3 identical spike subunits. So we've got here, they're shaded in all 3 different colors of green, or different shades of green. And it really does dot all around the outside of the virus. It is the most abundant protein on the outside of the virus. And if we go there now into a little bit more detail, there are really 2 heavily immunogenic regions on the spike protein. The first shaded here in cyan is what's called the N-terminal domain. The second, which is shaded here in yellow, is called the receptor binding domain. And actually this receptor binding domain is the most immunogenic portion of the spike protein. So it really elicits a very strong immune response, both through vaccination and natural infection. And then I just also wanted to point out somewhere around here, where you would basically have the top half of the spike protein and the bottom half of the spike protein meeting. This is where the cleavage site is for this protein. And this cleavage site really has shown its importance when compared to SARS-CoV-1, for example. SARS-CoV-1 does not have what we call a furin cleavage site. SARS-CoV-2 does, and that's really what we think is one of the main contributors to why this virus is so much more successful in infecting humans. But it's also a region that has really become prominent now with the emergence of the Delta variant, which has mutations in this region that we believe have made it more adapted to replicating in human cells. Sorry, so those are the little antibodies targeting those sites. So we're currently in crisis mode. This pandemic hit. The virus was way more transmissible than anyone initially thought it would be. And part of why this virus has really taken the world by storm is that most infections are asymptomatic and transmission occurs before symptoms develop. So yeah, so right now we're in this crisis mode of trying to build up immunity, trying to get as many people within the entire world vaccinated as possible to have some level of immunity, some level of protection. And I think this, you probably all are aware, this is just really, and it's unprecedented times. Never before have we seen vaccines becoming approved and coming into use as quickly as the vaccines for this virus have become available. But I also just want to temper that statement that, you know, a lot of the background work that has gone into making these vaccines has been years in the making. And a lot of the background work that has made having a SARS-CoV-2 vaccine possible in less than a year has been because of dedicated scientists globally who've been working on, for example, a vaccine to MERS coronavirus, to SARS coronavirus, even though they were not immediate large-scale threats. These were scientists with immense foresight who saw that coronaviruses really could be the next pandemic-causing virus, as we now see. The problem with this, with the world in general, is that we do not have equal vaccine coverage throughout the globe. So as you probably know, most of Africa, large parts of Asia have very, very low, relatively low vaccine coverage. But countries in the West, they have extremely high vaccine coverage. And so we really need to get a bit creative on how to spread out our doses here in Africa, in Asia as well, so that we can get as high coverage as possible, if not full coverage, but at least partial coverage. And then there's the question of durability. So once people are vaccinated, how long are their immune responses going to be protective? How long are they going to last before we need to boost? So this is data now from the 2 mRNA vaccine clinical trials. So the mRNA Moderna vaccine trial, Moderna vaccine is obviously not being rolled out in South Africa, but it is being used in other African countries. And then you all know the Pfizer-BioNTech mRNA vaccine, which is being used in our country. And both of these, from both of these trials, we see that there is quite long duration of antibody responses, up to 4 months at the time that this was tested. Of course, this might be longer as more and more time goes on. So that's quite short if you think about how long you would need before you needed to boost, but the data is still, the timing is still early, so this might be longer. Then if we look at the similar data for the Johnson & Johnson, for the advected vaccines, we see a similar kind of durability range of up to 4 months. And again, early days, so this might even be longer. Sorry, is my microphone off? No, it is on, okay. All right.
No, no, we can still hear you. It's fine.
Sorry, I just saw an old chat message and I got a bit confused. Okay, and then a really important thing that we're still trying to figure out is to define what is a correlative protection. So once people are vaccinated, what is the level of immune response? I'm going to say immune response because we don't know yet what type of immune response is protective necessarily. There is some early data here. This is showing a curve from a really good Nature Medicine paper where they basically did mathematical analysis of all the clinical trial data that's available for any vaccine against coronavirus. And they plotted here the titer of the neutralizing antibody response that was elicited by the vaccine against the efficacy of the vaccine. And you can see that the higher the titer that was elicited, the more protection you get. And so it certainly seems that neutralizing antibodies may be an important correlate of protection from infection. But the important thing to remember is that the immune system is not a single defined thing. It is a multifaceted, complex system that's interlinked. And so it's very unlikely that neutralizing antibodies are acting alone. And I think if we have any understanding of immunology, we know that there's the T cell and the B cell arm, and they definitely work hand in hand, they work together. And while neutralizing antibodies might be important for protection, some studies are showing that there is also a role for T cell help. And for T cells actually mediating protection very early on before antibodies have had a chance to develop. So then the next question is, does everybody respond equivalently? And I think this is especially important in South Africa, where we have such a large population of our people who are infected and living with HIV and AIDS. And so I think I don't need to say this to you. You probably are all very, very much aware of the stats, the HIV stats, but 19% HIV prevalence. So that's extremely high. This is some early data that's not yet peer-reviewed, showing the immune responses in HIV negative individuals and also in HIV positive individuals, people living with HIV. And these are looking now at the antibody responses against the entire spike protein, and then just that yellow region, if you recall, the receptor binding domain. And you can see that obviously at baseline, there's no responses, but at 28 days after administration of the vaccine, and then at 42 days, these responses look to be pretty equivalent against whole spike and similarly against just the RBD. But this is one vaccine. This is the ChAdOx vaccine. And we still have a lot of work in terms of looking at the others, looking at the mRNA vaccines, but also looking at individuals who have HIV and who are not virally suppressed, individuals who are not on ARVs. And so we still have a lot of work, especially in South Africa, in store for us to really evaluate whether individuals who are immunocompromised will be able to develop an immune response that is protective using the current vaccine regimens. And then I'm sure you've all also heard of this. Can we now mix and match? Can somebody get ChAdOx and then receive, as Angela Merkel did, the Moderna as her second shot? And certainly it seems to be the case. It seems to be that heterologous prime boosting is going to be a viable option. So this is now looking at data published by the Sander group, where you see in this part of the graph, individuals who received the approved and kind of clinical trial regulated method of prime and boosting with the same mRNA vaccine. And you can see there's quite a range in terms of titers following the prime, but after the boost, almost everyone has extremely high level of antibody titers. And then we see here where people then became received the prime with the Chadox vaccine. Again, a similar situation as what you see with the Pfizer, where you have this wide range in terms of responses, and then boosted then with the Pfizer. And you can see again, very, very high levels of antibody titers achieved. So it's looking like mixing and matching is going to be really good. It's going to be possible. And I think given, the shortages in vaccines, this is going to be increasingly important in countries like South Africa and other African countries. And then I'm sure, I actually think this was one of the questions in the Q&A, what about the new variants? How well do these vaccines work against new variants? So just to go one step back and talk a little bit about the mutations that arise in SARS-CoV-2. So if you recall that schematic from the first slide that I showed, this is the schematic of the genome starting at 1 and ending at about 30,000 bases. And each of these little lines that you see represents a mutation or mutations that have arisen in the genome. And you can see that there's mutations. This now is very old data. This was from December 2019 to April 2020. But already at that point, you can see that there were mutations that had arisen at almost every single site. Now, what's interesting to see is you have some areas where there's a lot of mutations that arise in just one position or one site. And then there's other areas where there are no mutations that have arisen. And this basically suggests to us that these kind of regions are functionally constrained. And so the virus really can't tolerate mutations. But in these other mutations, maybe it's not so bad, it's a bit more flexible and you get these mutations cropping up. Now, as with any other pathogen, bacterial, actually any other organism, mutations arise constantly. They're just a process of the life cycle and replication. Replication is not foolproof, never foolproof. And so you're always going to get these accidents or mistakes occurring. But to put it into context a little bit, SARS-CoV-2 mutates relatively slowly, especially when you compare it to something like HIV, something like influenza. It mutates pretty slowly. So to date, out of 30,000 genomes, there are about 60 mutations that are present in a large number of sequences. So there's 60 mutations that are actually being propagated, which is a small number when you think about it. So this here is showing that diversity that I was telling you about. And each of these rings here is representing a different period in time. So we have here at the center of the circle, the Wuhan virus, the original sequence that we have, and then all these other viruses have now diverged away from this with these blue, or I don't know, greeny blue dots belonging to the Delta variant. And you can see the Delta variant is very quickly replacing everything else. The important thing to note is that because there are so many individuals continually being infected with this virus, it presents the virus with many more opportunities to diversify and to become fitter. So most of the mutations that arise don't confer any advantage to the virus. But as we know in the case of Delta, there are mutations that do arise that make the virus better able to replicate in human cells, that escape from immune responses, and these all confer some kind of fitness advantage. So you probably are all aware there are now, because of the diversification and the impact this diversification has had, there are variants of concern, there are variants of interest, and there are variants under monitoring. So there are 4 variants of concern currently circulating in the world. The first that was identified was the Alpha variant. And you can see that spread quite widely. The second was the Beta variant, which I'm sure most of you are familiar with, and that was much more contained. It actually only spread to about, I think it's 85 countries. And then the third was the Gamma variant, which again was also very well contained, mostly to the Americas. But now we have the Delta variant. And actually this map needs a little bit of updating, but this variant has fast become the dominant variant globally. And the reason for this kind of dominance of these variants of concern is because each of them have had some or other selective advantage. In the case of the alpha variant, the beta variant, and the gamma variant, we know that all three of these shared the same 501YV2 mutation, which increased affinity for the human ACE2 receptor. But especially the beta and the gamma variants also had immune escape mutations. And then in contrast, the Delta variant, which also, of course, harbors immune escape, has become so successful because, as I mentioned, it has that furin cleavage site mutation that really increases its replicative fitness and therefore increases its transmissibility. So if we look at globally, this was 2020. We can see that there was, so unfortunately, the shading has changed. The shading of all these gray kind of circles represents all the older lineages that were circulating before the variants of concern emerged. And now in 2021, we can see quite a few variants of concern circulating, but in many countries, we see this green color dominating, which is basically the Delta variant completely taking over. And to put it into context in terms of South Africa, my slide is not moving. There we go. To put it into context, in terms of South Africa, we've seen the exact same thing. So in 2020, we had this multitude of lineages circulating in the country. And again, we've done the shading here according to old lineages that were circulating before the defining of variants of concern. And you can see there were many lineages circulating in our first wave of infection. And then in our second wave, at the end of 2020, we saw the beta variant cropping in, and that variant immediately began to replace everything else and dominate. And the reason was because it had a selective advantage. And then now in 2021, in our third wave of infection, we've seen the same thing where this beta variant and actually all other variants of concern, the alpha variant that was circulating at low frequency, has been replaced by the Delta variant, which is incredibly fit and incredibly transmissible. So what if we go now back to vaccines? What does this mean for vaccines? The continual emergence of variants with immune escape mutations, variants of increased transmissibility. What does this actually mean for vaccines? So if we look here at data that tested basically individuals who had been vaccinated with either the Pfizer or the AstraZeneca vaccines. So this Victoria strain is representative of the original virus and the immunogen that would have been used in the Pfizer vaccine and the AstraZeneca vaccine. You can see that pretty much all of these individuals have reduced neutralizing activity against the beta variant. So you're going from a mean titer of 1,105 to 146. That's a huge decrease in titer. Similarly with the AstraZeneca, a huge decrease in titer. And so while these variants now are much less sensitive to neutralization, there is still some activity. And so what this has shown us there is that potency is queen. So if you have a very potent neutralizing antibody response against any variant, you most likely will have protection. And as we've seen with the beta variant, as we've seen now with the Delta variant, unfortunately, protection from infection is no longer feasible, especially with vaccination using all the vaccines that are currently available to us that have their immunogen based on the original virus that was isolated from Wuhan. So this is not all bad news though. Even though we don't have protection from infection, what we do have is, and I think this is probably more important, is that we have protection from severe infection, in other words, from hospitalization and death. And that is key. If people are getting sick, getting a bit of a flu, getting some body aches, having to take a day off, That's much more manageable than having people dying from this virus. And so definitely protection is looking like more and more it's not possible. But this doesn't change the fact that if you don't get vaccinated, you have a much higher chance of dying from COVID-19 than if you were vaccinated. So in conclusion then, vaccines for coronavirus have been developed and deployed at unprecedented record speeds. But despite this, they have been well tested. They have been approved by many bodies globally, many health regulatory bodies globally. And because of this continual testing at rapid speed, we've been able to identify doses that are most successful at eliciting high responses, as well as the amount. The actual dose, how much to give somebody. So you may have heard discussions currently about really how to stretch out our doses in South Africa, given how limited we have and how many people we still have to vaccinate. And there's even suggestions of boosting, for example, our healthcare workers who for the most part receive the one dose Johnson & Johnson vaccine, boosting these individuals with a half dose or a quarter dose of the Pfizer vaccine, simply because this has now been tested and has been shown to be as good as a full dose. In terms of South Africa and other parts of Asia and Africa, we do need to do more investigation into how these vaccines elicit responses in HIV positive, viremic or immunosuppressed individuals. Do we need higher doses? Do we need more frequent boosting, for example? We also know that the mix and matching strategies realistically are unavoidable, but that they are very good, that they're as good as using a homologous prime boost situation. And they might actually be what is feasible and what is achievable considering the vaccine shortages. And then the last point that I made is that high titer responses are the best solution to combating new variants, but also to slowing the emergence of other variants. And so with that, I would just like to extend many, many acknowledgements to multiple individuals at the NICD who've contributed to some of the data that I presented, but who've also contributed to many discussions and a lot of thinking around this. Individuals from the Center for Respiratory Diseases and Meningitis, from the Center for HIV and STIs, as well as our leadership, of course. And we have within the NICD a couple of COVID and SARS-CoV-2 focused groups that really have been instrumental in driving discussion. And then I would also like to thank the Network for Genomic Surveillance in South Africa, which is the really collaborative body of individuals and institutes who are working together, well, have been working together since last year and continue to work together to really do excellent genomic surveillance of this virus on a global standard. With that, I would like to say thank you, and we can open up to the Q&A session.
Thank you very much, Dr. Biman, for this wonderful presentation. I think it's something that we all appreciate. And as we can deduce from this presentation, it's not just an academic exercise. She has given us information for potential and real solutions to a current global challenge that we all have. So this is very good. So for the question and answer session, the first thing we need to do is to address the questions that were posted online. Dr. Biman, you have those questions, so you can just address those questions, please.
All right, so the first question comes from a 17-year-old who is fascinated by microbiology. So she or he wants to know, why can the normal vaccines like Johnson & Johnson or Pfizer not be administered to children? And why does it have to be approved for children to be able to get the jab? So I'm not sure when the questions were posted, but as of the 10th of September, the South African health regulatory body, SAHPRA, has approved the Pfizer vaccine for use in children 12 years and older. So initially, these vaccines were not approved for use in children, simply because all the clinical trial data that had been gathered had been done in adults. There had not been any safety testing specifically done or efficacy testing specifically done in children. Since then, that has changed. The US really has been the driver in this, but also Germany, I think, and France have also done some studies looking at how children respond to these vaccines. And so the US FDA was the first, I think, to approve the Moderna and Pfizer vaccines to be used in children. And so, like I said, as of the 10th of September, we can now we are allowed to use the Pfizer vaccine in children. And yeah, it has to be approved simply because of safety. Safety has to first be tested. So the way that these clinical or the way that vaccine approval happens is you've got to have first a phase 1 clinical trial, which is really small number of individuals. And in that kind of a trial, all you're looking for is safety. You're making sure that nobody has serious adverse effects, that there's nothing, no red flags or those kind of warnings. Then you move on to a phase 2 trial, which is a larger trial, again, looking mostly at safety. And then you move on to a phase 3 clinical trial, which is enormous, thousands and thousands of individuals, generally within multiple global regions, so that you can look at how the vaccine performs in different populations. And there you're looking, obviously, you're always continuing to check for safety, but there you're actually looking for efficacy to see whether the vaccine works in what it's supposed to do. And after a vaccine passes phase 3 clinical trials and it is actually efficacious and safe, it has to be approved, and not everything gets approved. And the approval is really country specific. Just because a vaccine is approved, for example, in the US, does not mean it will be used in South Africa. So it is all dependent on the country's health regulations, and the regulatory body within that country will then approve or not approve certain vaccines. And once it's approved, that's when it then can become used, can be fully authorized, in other words, and can be used. So I hope that explains that a little bit, that now we have approval in children just for the Pfizer vaccine here in South Africa. Okay, the next question then is, the Pfizer vaccine is now approved for use in adults. How does the vaccine development and approval differ for young children? Is it necessary for them to get vaccine? I think I've already answered that question. The approval for young children is the same, but typically a vaccine is tested out and approved in adults first before it's considered for use in children and before it is tested out in children. Is it necessary to get vaccinated? I think now more than ever, it is critical that children, if possible, can get vaccinated. The reason for this, I'm not sure if any of you have seen, but again, thanks to the Delta variant, we're seeing many more infections overall. We're seeing, of course, many more infections in children, and we're seeing many more cases of pediatric admissions because of COVID. And this is quite different to what we had seen with, for example, the Alpha, the Beta, the Gamma, and also the original viruses. So it is a good idea for them to get vaccinated. However, the big disclaimer is that when younger people are infected with SARS-CoV-2, they tend to handle it a lot better than if older people are infected with this virus. We know that there is this age discrepancy where older people are more likely to end up in hospital and end up dying from SARS-CoV-2 infection. And so in terms of South Africa, right now, our priorities are to vaccinate older people, they will get priority. Our priorities are to vaccinate healthcare workers, essential workers, and of course, people with comorbidities. So this is, in South Africa, this is still under discussion, whether, when, and how to roll out to children. But in my opinion, I think it is necessary for them to get vaccinated, really to give everyone a chance at not having severe infection. The third question then is, do any of the vaccines contain the luciferase enzyme, and what is the role? As far as I know, none of the vaccines contain the luciferase enzyme. So that is an easy question. But this may be, I don't know, related to some other lab papers maybe that this person read. So the luciferase enzyme, for example, is used in laboratory studies when kind of pseudoviruses or fake viruses are grown up of SARS-CoV-2. And generally you use a luciferase enzyme and you use the luciferase substrate in conjunction with the enzyme that's produced by the pseudovirus to test whether there was any virus, whether there was infection. But this is all within cell culture in a laboratory. This is nothing of the sort is injected into humans. What are the components in COVID vaccines? So there are a couple of different platforms, shall we call them? So as you probably know, there's the mRNA vaccine, there's the ad-vectored vaccines, and there's the inactivated vaccines. So those three differ. The mRNA vaccine is RNA that you inject into the muscle. And when that vaccine gets into the cell, that RNA is the instructions that's given to the cell to tell the cell to make the spike protein of the coronavirus. Same thing with the ad-vectored vaccine. Again, you're putting in the instructions, and then the instructions tell the cell how to make that protein, how to make that coronavirus protein. It's just one protein of the entire genome. If you remember that schematic, the S protein, it's just that protein. And then the body sees this S protein, it recognizes it as foreign, and it starts making an immune response to it. The inactivated vaccines, on the other hand, are the actual whole vaccine that has been destroyed. So it's not able to replicate. It then is injected. And again, because it is a virus, the body recognizes that as foreign and produces an immune response to it. Then in addition to the actual immunogens, there are also stabilizers. There are things that maintain the pH, because obviously you have to maintain a certain pH. You have to maintain that because otherwise your immunogen is going to fall apart. And then there's also always an adjuvant. And an adjuvant is basically something to make your body respond better, to make your body realize, hey, there is something here, and make the immune response stronger. Those are typically the main components in vaccines. All right, so then the next question is, are there any side effects or adverse health implications associated with the vaccine? So yes, there are side effects, and it really does vary by person. You can have somebody getting vaccinated and feel perfectly fine the next day, have nothing, not even a sore arm where they received the vaccine. And then you can have other people who might develop a fever, who might have body aches. And I'm one of those people. I was down for like 39 hours after I got the vaccine. Those are kind of side effects, but it's not severe and it's not bad. It's manageable. The adverse events that have been reported, I think mostly for the Chaddock, and the Johnson & Johnson trials, have to do with clotting, which I'm sure most of you have heard about. But this is really it's specific. It's not general to the population. So this is really a 1 in a million chance of it happening. And I think if you actually get COVID, your chances are much higher of getting clotting. So the benefits outweigh the risks. And then also it's generally linked to people who have some kind of blood condition. It's not the general population who are fine and who, don't have any health complications. Then the next question is, there is information going around that the vaccines contain graphene oxide. This is not true. The vaccines do not contain graphene oxide. If you are really interested to know what are the individual components of each vaccine, just go to the US FDA website. They, by law in the USA, are required to list every single ingredient in any medically authorized therapeutic or vaccine. And so they've done that. So if you're really curious, you can go ahead, go to the FDA website and download the information sheets. They will have everything listed. Graphene oxide is not one of them. So the next question also relates to that. So I'm not going to go over that. Then it says, if people are vaccinated, why and how would the unvaccinated people be a health threat to the vaccinated people? So as I mentioned, now with the emergence of new variants, protection from infection, even after vaccination, is looking less and less likely. And so people who are unvaccinated, number one, they are going to have no immune response to this virus. So when the virus infects them, even if they don't have symptoms, they could have a very high viral load, which means they have a much higher chance of transmitting the virus. And that's really how they're that's how they're causing a health threat to people who are vaccinated, because even if you are vaccinated, you're still susceptible to infection. Whereas on the other hand, if these people themselves also became vaccinated, their body already has memory, their body already knows how to respond to this virus. And so they will have much lower viral loads and will have much less chance of transmitting the virus. Okay, so the next question then is, what would be the benefit of this vaccine at this stage if they aren't stopping infection and transmission? And this is a really important question, which I hope I have already answered, but I think is important enough to answer again. Because you don't get protection from infection with the vaccine, it doesn't mean it's useless. It's extremely crucial because it still has very high levels of effectiveness in terms of preventing you from becoming seriously ill and from being hospitalized. Now, especially in the case where we have our waves and we have lots and lots of infections, you really don't want to be in hospital because number one, our hospitals become overwhelmed. Number two, there are not enough ICU beds, for example. There's not enough oxygen. And so your chances of dying are much higher. So that is the benefit. Get the vaccine so that you don't actually get severe illness. Get the vaccine so that you don't die. It's not about protecting others anymore. It's about protecting yourself from death. All right, then the next question, what are the mutations and will existing vaccines still be effective? So I think I've touched on this already. I'm not going to list the actual mutations because there are plenty. If you're really curious, there's a wonderful site called outbreak.info, and you can pull up every single variant of concern, every single variant of interest, even if they're not a variant of concern, every single lineage, and it will show you graphically quite nicely what the different mutations are in each of the different genes. If you're really interested, that's where you can go have a look. And I think I've already mentioned that, yes, the vaccines are effective against these variants in terms of preventing hospitalization and death, but not in terms of preventing infection. And I think that's also an important thing that we all need to remember. And that's why we need to still continue to wear our masks when we're in public spaces, why we still need to sanitize and practice all the normal COVID protocols. All right, so in the next question, in your opinion, what is the end goal of COVID vaccines? Are we hoping to eradicate the virus, or will it become seasonal like flu? I think that it is very unlikely that we will eradicate this virus. There's multiple reasons for that. Number one, it spreads like wildfire it has spread like wildfire through our human population. It can cause reinfections. And number three, there is so much spillback into other animal reservoirs. So this virus is very promiscuous. It doesn't only infect humans. It infects cats, it infects dogs, it infects minks, it infects rats, mice. And And so because it's now infected so many different populations, I think it's very unlikely that we will be able to eradicate it. In terms of whether this will become a seasonal thing and whether we will need to get a flu shot, a coronavirus shot every year, I really am not certain on that front. I think at present, it looks like we will need to, especially in our elderly population, especially even maybe in our healthcare workers, we may need to give boosters. But in terms of the general public, I'm not really sure. But even for influenza, you know that in South Africa, we always have limited influenza vaccine stock. So we do prioritize our elderly, we do prioritize pregnant women, we do prioritize HIV positive individuals. So it might be a similar situation with the coronavirus vaccine. I think what we're hoping for, and I kind of alluded to this, is that because the coronavirus does not change as drastically as flu, does not change as drastically as HIV, we're hoping that with enough boosts of different variants, we might be able to elicit cross-protective immune responses, and we wouldn't need to be boosted. Okay, then adults and children get vaccinated against a variety of illnesses such as polio, MMR, tetanus, etc. But still a lot of people are opting out for COVID vaccines. What do you think is the biggest driver for vaccine hesitancy? This is not a scientific question. This is an opinion question. But I do think that the biggest driver is fear of the unknown. And when people don't understand, that makes them hesitant. I do think that's a big driver, and that's really from personal experience. Speaking to people who do not want to take the vaccine, they have concerns that they are going to get sick, that this vaccine will interfere with other medication that they may be on. That's what I think might be the biggest reason. But yeah, if anybody else wants to chip in and give other reasons, please go ahead. Okay, then we've got a couple more. Number 13 is there are a lot of theories about how COVID started, such as the lab leak theory. What does the genome of the virus tell us of its origin? So there's a brilliant report that was put together by the WHO. I think one of the authors is Peter Daszak, there's Marion Koopmans, a lot of Chinese scientists. I think if you are really, really interested in the details of this, you should read that report because it is excellent. It provides a lot of evidence for why the lab leak theory is not realistic. So the first thing about this coronavirus, right? It is most similar to a bat coronavirus that was identified from a lab in China, a lab that specializes on coronaviruses. Now, before the SARS-CoV-2 emerged, the only sequence they had from that rat virus was of the spike protein. They didn't have the full genome. They didn't even know what the rest of that virus looked like. Since then, they filled it in. That is its closest relative. I think it is 96% similar. That bat virus is called rat G13. That's its closest relative, 96% similarity. If you compare the genome of humans and chimpanzees, we are 99-point-something% similar. So those two viruses are more different from each other than we are to chimps. So there in itself, that's the closest relative that we know of. And there in itself, it's very distinct, it's very different. The second thing in the genome is that, and I've mentioned this a couple of times, it has this furin cleavage site. And this, I mentioned as well, is what has distinguished SARS-CoV-1 from SARS-CoV-2. This is what makes SARS-CoV-2 so much more adapted to human cells. This is why it's so successful at infecting us. This furin cleavage site is not uncommon. It's found in other coronaviruses that infect other animals. And so coronaviruses do this all the time. They recombine. So in other words, a rat coronavirus would steal a portion from a pig coronavirus, for example, and then they'd make a new coronavirus. They do this all the time. So this sharing of parts of the virus happens all the time. That's another kind of big pointer as to say that this was really something that arose through in nature. In addition,
Sorry,
if we
Dr.
look,
Bina, sorry to interrupt.
have we run out of time?
We are running out of time.
There's a lot of questions.
There are a lot of questions. So, okay, now I think the approach we'll take is we would be able to send you all the remaining questions. And what we'll do now is just to take one extra question from the audience, from people who are listening now from the floor. So anybody that wants to ask any question, just one question from the floor. You can either raise your hand or just speak, it's fine. Any question from the floor? So there are many other questions here. Okay, if there is no other question that if people don't want to speak, then just a question for those recently now. What are your thoughts on ivermectin? That's medication that people are talking about. What are your thoughts on it, Doctor?
So the clinical studies that have been done do not show any efficacy of ivermectin, so it does not work. It is a non-specific drug that's used to treat other pathogens. I think it's helminths, if I'm not mistaken. And so there was really this hope that it would be useful, but the clinical data shows otherwise.
Okay, that's fine. That's fine. So just one more or two more. Can you compare the success of HIV and COVID-19 vaccines? What are the challenges in making these vaccines?
So we don't have an HIV vaccine to date. HIV is, like I said, far more diverse. It mutates at a much faster speed, and that's part of the reason why it's so challenging to make an HIV vaccine. So I think the mutation rate probably is the biggest challenge as to why we aren't able to make an HIV vaccine, but why it has been so simple and easy to do this for coronavirus.
So the other questions that are here, I think you've answered most of them. Just one more. Can you possibly explain why there is where we have 2 doses versus 1 dose?
Sorry, I didn't see that question.
So where I think the person is referring to the fact that in some situations you have 2 doses. For instance, Pfizer, and for Johnson & Johnson, you just have 1 dose.
So, I mean, in all honesty, I think it would be best if everyone could get 2 doses. With the Johnson & Johnson 1 dose, you don't get as high titers as you do with 2 doses with the Pfizer, but that's just how those vaccine regimens were designed and how they were tested. So that's why you have the 1 versus 2 doses. But in all honesty, I do think 2 doses and a boost is better.
Okay, so just to wrap up, Dr. Izel Mufandu, you wanted to say something quickly?
Yes, thank you, Prof. Rashid. Thanks a lot, Dr. Gina, for a brilliant talk. I think we need to just address as a round off this question, and I think it's pertinent and probably will answer a whole lot of others on the platform. Why is the injection called a vaccine if it doesn't give protection against infection?
So there are multiple types of vaccines. Vaccines do not only prevent infection. You can also have therapeutic vaccines, which actually this probably is more likely to be. It's something that's going to help you combat the illness. So yeah, so there isn't just one type of vaccine. Vaccines are not only preventative. There are both preventative and therapeutic vaccines.
Okay. Perhaps rabies would be a good example of the example that you're talking about, the rabies. Okay, yeah. Okay, thank you very much for your time, for the presentation. And I think we are all enlightened now. So I would now give the floor to Prof. Carlos Benzidel, our unit director, to give the vote of thanks. Thank you.
Thank you, Prof. Rashid. Good morning, everyone. There were about 49 participants during the course of this webinar, and there's a huge number of individuals that are watching on YouTube. Greetings to all of you. This is Heritage Month, and as Prof. Madupe has indicated, microbiology as a science has a very long history. It was only recently that the International Microbiology Day was instituted. So celebrating that is absolutely important, particularly from the Northwest University's point of view. So from the Faculty of Natural and Agricultural Sciences, the Unit for Environmental Sciences and Management, Food and Security and Safety, the School of Biological Sciences and Microbiology, both the Potchefstroom and Mahikeng campus. Thank you to everyone that participated in this webinar. Prof. Rodney, the Deputy Dean for Community Engagement, thank you very much for your opening remarks and for your welcome address. Dr. Lesego and Dr. Hazel Mafundu, the Chairperson and Deputy Chairperson of Microbiology. Thank you for organizing this event. Prof. Sarina Claessens and Prof. Ozenil Ribitso, the Director and Deputy Director. Thank you very much for allowing the organizers to have this event, to put it together. The event was funded by Microbiology and NRF WhatNet. WhatNet is a collaboration between the Northwest University and university in Germany, and funding for this event came from that. Dr. Janelle Biman, thank you very much for an inspirational talk, for the ability to simplify the complex scientific concepts within the evolution of this very important virus in this time, at this stage in the history of mankind. Thank you very much for that, but also that you kept the attention of microbiologists throughout your talk. Thank you for the excellent manner in which you responded to the various questions. Prof. Rashid, thank you for your role as Program Director. Thank you for all the participants in engaging the questions. There was a team of that organized this event, and I'd like to record their names for noting. Dr. Lesego Molaletom, Dr. Hazel Mafundu, Dr. Gaugelo Lakota, Mr. Glenville Fransman, Mr. Abram Matlatsi, Ms. Raiza Biko, Ms. Lee Shanaka, Mr. Ruan Fori, Ms. Janae Beekes, Ms. Sepati Pupedi, and Mr. Berno van Niekerk. Then the providers of this platform, New Sound Productions, Paul and Massimo, thank you very much for professional support that you have provided. The platform was absolutely wonderful, the preparations as well as the event this morning. So thank you very much to everyone that participated in this event. Thank you.
Thank you, everyone. So that will be the end of the event. Bye.
Bye-bye.
Bye-bye.